The overall goal of the following experiment is to test for specific variants in the BRCA1 protein using an in vitro homologous recombination assay. This is achieved by replacing the endogenous wild-type BRCA1 protein with the variant being tested through the transfection of both short interfering RNA specific to BRCA1 and plasmid for re-expressing test BRCA1 variant proteins into cells. As a second step, the short interfering RNA and the plasmid for expressing mutant BRCA1 is retransfected in addition to a plasmid for expressing the ISCE1 protein, which causes DNA damage at a specific site in the DNA genome of the test cell. Next, the cells are incubated for 2 to 3 days in order to allow for the DNA damage and repair to occur. Results are obtained that show whether specific protein variants of the BRCA1 protein function in DNA repair by homologous recombination based on flow cytometry analysis of GFP-expressing cells.
This technique combines two established methods in order to create a versatile system for analyzing specific protein variants in the repair of DNA damage by homologous recombination. The main advantage of this technique over existing methods is that previously established methods relied on cell lines that had a genetic knockout of BRCA1. We use a more manageable cell line derived from HeLa cells that enables BRCA1 depletion by RNA interference of the endogenous protein and add back of test protein. In these cells now expressing a variant BRCA1 protein, we can test for homologous recombination.
Generally, individuals new to this method will struggle because of excessive cell death during the procedure. It is important to have a transfection condition carefully set, maximizing the effective recombination reaction while limiting cytotoxicity.
Prior to the start of this protocol, the HeLa DR13-9 cell line was established by transfecting HeLa cells with PDR-GFP. Maintain this cell line on 10 cm Petri dishes in purimycin for selection of stably transfected cells. The recombination substrate in the PDR-GFP plasmid and in the stable transformants contains two inactive alleles of GFP. One allele is inactive due to the inclusion of the 18 base pair restriction enzyme recognition site for ISCE1 in its coding sequence. Expression of ISCE1 in these cells creates a double-stranded DNA break, or DSB. On the second allele of inactive GFP, the portion of its sequence correlated with the ISCE1 site on the first allele is wild type. This second GFP allele can function as a sequence donor for homologous recombination repair of the DSB and would result in an active GFP allele, which is readily detected by flow cytometry. The day before the first transfection, obtain a 10 cm dish of HeLa DR13-9 cells that are at least 90% confluent. Trypsinize the cells in 1 ml for 2 to 3 minutes. Stop the reaction by adding 9 ml of DMEM-FBS and mix the cell suspension well by pipetting. Add 40 microliters of trypsinized cells per well of a 24-well plate in 0.5 ml of DMEM-FBS. Incubate the cells at 37 degrees Celsius and 5% carbon dioxide overnight. On day 1, cells should be between 40 and 50% confluent. Begin transfection by mixing both the siRNA to deplete endogenous BRCA1 and the BRCA1 mutant-expressing plasmid in OptimM media. Then, combine the nucleic acids with lipofectamine in OptimM media. Proceed with the lipofectamine transfection as per the manufacturer's instructions. Following transfection, return the cells to incubate. Replace the media 4 to 6 hours later with medium that does not contain antibiotics. Return the cells to incubate overnight. On the second day, trypsinize the cells by adding 0.3 ml of Trip-LE trypsin reagent to each well. After a brief incubation at 37 degrees Celsius, loosen the cells by pipetting the cells up and down in the trypsin reagent. Once trypsinized, transfer the cells from the 24-well plate to a 6-well plate containing 2 ml of DMEM-FBS to halt trypsin activity. Allow the 6-well plate to incubate overnight under the same conditions as before. 2 days from the initial transfection, prepare the siRNA, the BRCA1 mutant-expressing plasmid DNA, and the ISCE1 expressing plasmid in 62.5 microliters of OptimM. Mix this solution with 2.5 microliters of lipofectamine 2000 in 62.5 microliters of Optimem. Proceed to transfect the cells. After a 4 to 6 hour incubation, replace the media with DMEM FPS lacking antibiotics. Return the cells to the incubator. On day 6, trypsinize the cells in each well by adding 0.5 milliliters of TRP-LE trypsin reagent. Stop the reaction by adding 2 milliliters of DMEM FPS. Collect the cells by centrifugation at 1000 RPM for 5 minutes at room temperature. Resuspend the cells in 1 milliliter of fresh DMEM FPS. Analyze 10,000 cells per well for GFP expression by flow cytometry. Single live cells are gated using forward and side scatter parameters. GFP-positive cells are directed using the GFP fluorescence detector, and results are presented as a histogram. Remaining cells can be replated for photography by fluorescence microscopy if desired. BRCA1 regulates the pathway for repair of double-stranded DNA breaks by homologous recombination. Those cells which have undergone repair by homologous recombination are readily detected by fluorescence microscopy. Depletion of BRCA1 by RNAi results in a sharp decrease in the detected GFP-positive cells. The output from the FACS caliber is a histogram with GFP intensity per cell on the x-axis and the number of cells on the y-axis. In the absence of transfected ISCE1 expressing plasmid, there are no GFP-positive cells detected. The results in columns 2 through 6 were all taken from cells in which ISCE1 expressing plasmid was transfected on day 3. In a typical set of results from a single experiment, 15.5% of the cells are GFP positive after ISCE1 expression and control RNAi depletion for an irrelevant gene. By comparison, depletion of BRCA1 and add-back of an empty plasmid vector reduced GFP conversion to 0.7%. Addition of exogenous wild-type BRCA1 fully restored homologous recombination as detected by obtaining 15 to 16% GFP-positive cells. Similarly, expression of another variant of unknown significance, the BRCA1 I21V mutation, fully restored BRCA1-dependent homologous recombination function. Add-back of the BRCA1 M18T mutant had a similar effect as vector control. Since this variant expressed at similar levels as the endogenous protein, it suggests that the BRCA1 M18T variant is non-functional in homologous recombination.
While attempting this procedure, it is important to remember to monitor the growth of the cells. If the cells are growing slowly and at low confluency, then the amount of transfection mixture used must be reduced. It is also important to remove the transfection medium in a timely fashion. We find that the days following transfection, there is a high amount of cytotoxicity if antibiotics are included in the medium. For this reason, all the medium after day 1 lack antibiotics.
After its development, this technique paved the way for researchers in the field of breast cancer research to explore the genetics of BRCA1 variants with unknown clinical significance in cancer clinics. We hope that the functional BRCA1 assay developed in this work will enable improved prediction of the effects of BRCA1 variants on cancer predisposition.